Thank you. So I was asked to sort of look back at things we've been doing over the past few years, as well as the things that we have in the pipeline and the new activities that we're planning. So this slide just sort of summarizes where we began our adventure in ME. This is back in 2015, when we were the Institute of Food Research. And because I ran a program very interested in gut biology, I was intrigued by the connectivity or the association of GI disorders with ME. So we decided that we'd sort of look in the gut and see if there's any evidence of a more causative association. And so we started off by asking two questions, the first of which I think has been fairly convincingly answered, particularly by people like Mady and Maureen's groups and our own group. So there clearly is something abnormal about the microbiota, the gut microbes. Why and how, of course, remains an unanswered question, but clearly there's something different. The second question we're getting closer and closer to answer, and that is, are gut microbes the trigger that causes an autoimmune-like disorder in ME? So that is ongoing, and I hope maybe next year we can report back on some of those findings. So this is where we are now from 2019, this year, the Quadra Institute, which is a bit of an unusual institution for the UK in that it has an NHS department in the basement, that's an endoscopy unit, and then above it, 2 floors of research space. And the overarching, so the mission statement is to understand how food and gut microbes keep us healthy and also prevent us from getting diseases, particularly on diseases associated with diet and those with age-associated conditions. So in terms of the sort of suitability of the Institute, I think it's well placed for addressing ME and changes in the GI microbiota. So I put this slide up just to sort of familiarize yourself, remind you of what we know about the human microbiome. Most of the information on the text slide here, this sort of relates to looking at the bacteria in the gut. And I think it's fair to say about 90% of all microbiome studies are focused on the bacteria. But they only make up one constituent of the human microbiome. There are, in fact, more viruses than bacteria, particularly in the GI tract. There are also fungi, protozoa as well, and some archaea, which are the sort of ancient bacteria, if you like. And what we're doing at the Quadram is trying to look at the microbiome as a complex microbial community. So we're looking at how these different constituents interact to provide health benefits or cause disease in some situations. So we've begun, as where most people have, by looking at the bacteria, but we're now making advances in viruses, and we're starting to develop and refine our protocols for looking at fungi, particularly yeast, in the GI tract and how they may relate to health and disease. So this is sort of the paradigm for linking gut microbes to the central nervous system and the brain, and this is related to dementia. So there's evidence here linking gut microbes to the development of Alzheimer's disease that comes about through various stresses, environmental, medication, just through aging, and how this can cause changes in the microbiota, called dysbiosis. This can lead to disruption of the gut barrier, leakiness, if you like, leaky gut. This then triggers an immune response which spreads through the body, and this can result in changes in the CNS and beta-amyloid disposition. For Parkinson's disease, it's somewhat similar, although there's an interesting observation that some microbes can produce this alpha-synuclein, which accumulates in the brain to cause Parkinson's disease. So maybe there's a more direct association with gut microbes in Parkinson's disease in producing the protein that causes the pathology in the brain. So we were interested in ME from this similar perspective, and I don't need to go through this with you, I'm sure, but we chose to focus on the severe affected ME patients, thinking that the phenotype or the appearance or the manifestation of the disease, biomarkers will be more clearly defined, we could identify them, and there's less ambiguity about diagnosis. So we're looking at patients that are essentially housebound, or in extreme cases, bedbound, unable to tolerate any noise, light sensitivity as well, and they need daily and almost 24/7 care to keep them going. And so it's surprising that severe ME patients are sort of if you like, an ignored contribution to the research. There's very few studies that have been done to date that focus on the severely affected ME patients. As you can see here, there's really little evidence of that. I could find one example. This is the FINE trial, which some of you may be familiar with. This pragmatic rehabilitation, where patients were visited in their home to try and rehabilitate them. As you can see, the studies there indicate that they're either ineffective or there's no cost benefit to this. So other than this, I don't think there's been really much in the way of trying to understand the pathogenesis, the cause of disease in severe ME, which is where we began our studies. So these are the people in my group. I think it's encouraging to see that, unlike me, they're very young, very vibrant, very enthusiastic, and one is at the back there, Daniel, who I'm sure you know. Daniel is now Dr. Daniel, having gone through the process of his PhD. And we have Ernie, Fiona, and Catherine. And then the more sort of established, older components of the team here, Tom Wildman, Amalak, who is our link to the ME service at St. Helier, and Leslie, who's helping us with a lot of the virome studies. So this is sort of where we began. So we were particularly interested in looking at severely affected individuals, and we wanted to recruit also their carer or their parent, because one of the major confounders or causes of variability in the microbiome is the environment and lifestyle. So we wanted to try and control for that by having control individuals that lived in the same household, ate the same things, probably not in the same amounts, so we could control for a lot of that. So as you can see, we're able to recruit individuals and controls in the same household. Some controls were reluctant to join the study, despite the patient being very happy to work with us. So we ended up having to sort of stretch beyond Norwich and beyond London, actually, to recruit these patients. And in some weeks, we'd have to travel maybe 1,000 miles to collect and visit the patients. Obviously, they cannot come to the hospital. We have to go to them to collect their samples. This is the device we started with. It's a Dutch device. It sits on the toilet. We're given a sachet to keep oxygen away from a lot of the microbes in the sample. That's important because most of the microbes in the stool are killed by oxygen exposure. They were stored in the fridge for a short period of time. We collected them, took them back, and we processed them to look at what's in the sort of mass, this hard mass of this material, the microbes, as well as the more liquid phase to look at metabolites in these samples. So Daniel is the person who sort of initiated this, and the question we posed, or the hypothesis, is that severe ME patients would have a distinct profile of microbes that reflected the severity of their clinical condition. And that's when comparing them to individuals that live in the same household. So this is a bubble plot which summarizes about 4 years of Daniel's work. It's a bit disappointing, it's only on one slide, but never mind. So what you can see is that sort of the size and the colors illustrate the abundance and the presence. So the bigger the bubble, the more there are of that individual. You can see here we've got the ME patients, and on this side we've got the controls. And you can see straight away there's some differences, but also there are differences within each group as well. And this comes back to the individual nature of everybody's microbiome. This is very in-depth sequencing, so we're looking at very small populations of microbes that make up the whole microbiota. But you can see it reflects very well the individual uniqueness of an individual microbiota. But despite that, there are some commonalities. So this one here, in the controls of Rumnococci, there's a Fecal bacterium here, and there's a Bacteroides down here. So there are some commonalities and there's some distinctions. So the next slide just summarizes all of this analysis. And what we identified was what we call a core microbiome. So these are species of bacteria present in all individuals within our control group or our severe ME. And you can see by just comparing these boxes that they're distinct. And the three things that sort of jumped out to us were these three organisms here, Agaricella, Oxalibacter, and Faecalibacterium. And these are interesting because they've already been associated with certain diseases. So Faecalibacterium in particular is an interesting one because this is, I think, one of our sort of beneficial microbes and a key indicator of having a healthy microbiome. It's been used to develop probiotic-based therapies to treat patients with inflammatory bowel disease. So there's a lot of interest in developing this. And changes in the makeup of the size of this population have been correlated with various diseases, including obesity, asthma, and depression. Then the others are more unusual bacteria. We don't really know too much about them, but again, there are some clear disease associations. Now, this Faecalibacterium associated particularly interested because it correlates very well with the findings from Mady's group as well as Maureen's group. So here they have also shown that there's a decrease in this Faecalibacterium praesenutzi. So this might be one of a useful indicator of a healthy microbiome, or its absence indicative of a dysbiotic microbiome. However, it is difficult to compare our results with these studies, and in fact to compare these different studies, because they all use a different methodology, different processes, different types of analysis, different sequencing platforms, etc. So that does make direct study-to-study comparison difficult. And this is particularly of concern to the International Human Microbiome Consortium, of which the Quadra is the UK representative. And this group of individuals, which is across the world, is really coming together to try and standardize protocols for looking at the microbiome. So we're trying to develop the process by which people across different countries that want to look at microbiome can use the same protocols, same procedures, so it makes comparison of the data sets at the end much easier to do and more valuable. So we've moved on, as I said, to looking at viruses as well, and this is the work of Ernie and Fiona. And viruses are interesting because there are not just one type of virus in the gut, there are at least 4 different types. There's archaeoviruses, eukaryotic viruses, which infects the cells of our body, and then there's these virus-derived genetic elements. By far the most prominent are bacteriophages, and these are viruses that infect bacteria. And these are present in the gut in vast numbers, and they alone can outnumber bacterial cells by about 5 to 1. So most of the microbes in your gut are actually viruses, not bacteria. And they clearly can shift and cause changes in the gut bacteria populations, which could be beneficial or detrimental. They're highly individualized, just like your bacterial populations are. Your virome is distinct as well. And not surprisingly, changes in the virome have been associated with inflammatory bowel disease and diabetes, and we believe also in ME. And so bacteriophages are in fact probably the most abundant living entity on the planet. Most of them are found in the oceans and the soil, but the GI tract does contain a lot, a quadrillion, I believe, 10 to the power of 24. And we are in fact taking in a large number of phages every day, mostly through the things we eat or drink. And we are in fact, at the other end, we're shedding quite a lot of viruses as well. So we have this continual trafficking of viruses through the body, some which will stay in the body, others that will just pass through. And this is important in terms of trying to identify what viruses are really present normally and stay in the gut versus those that are just passing through. Okay, so the questions that we've asked here relate to looking at these bacteriophages. So what types of viruses are present in ME patients? Are they different from those that have less severely affected patients? And are these viruses pathogenic? Can they kill bacteria and explain the dysbiosis that we see in ME? And more importantly, are they a trigger for the immune response, which we think is part of the pathology of ME? So these are the questions that we've been particularly focusing on. Unlike looking at bacteria, looking at viruses is much harder to do because there are no real standardized protocols. So the reference databases that you use to identify viruses is very, very small compared to identifying bacteria. So we can isolate virus sequences, but we can't identify them because we have no reference to compare them to. And also, there is no standard protocol for isolating viruses from human stool samples. And then beyond that, once you have the sequences, there's no specified sort of pipeline to analyze your data and to then produce the results. So we've had to sort of invent a lot of this, if you like, really starting from scratch. And so this is a very simplified cartoon of the main process through which we process a stool sample. We filter it to try and remove most of the bacteria. We then centrifuge it to try and pellet the viruses. Sometimes we precipitate them using chemicals. This gives us then an enriched fraction of viruses from which we can then extract their DNA, sequence them, and then analyze the sequences, trying to identify what these viruses are. So these are some of the viruses. This is what they look like from an ME patient. As you can see, they're morphologically distinct types. Some that just seem to have the typical head shape. Others have these incredibly long tails. Others are here as well. So it's the tails that allow them to attach to the bacterial cell that then they can invade. So that these sort of structures are very important to the virus. So different types of viruses. And the result of a lot of the analysis is that we think we've identified the most prominent viruses present in ME patients, although we can only identify 25% of the viruses isolated because we don't have the databases and the reference genomes to identify them. But the most prominent ones are these Podoviridae. So these have the shortened tails. And this does sort of contrast with results that Maureen published a while ago, where she described the prominence of the other two types of viruses there. Again, this may reflect differences in protocols, but also looking at severe versus less affected ME patients. Now, at the bottom here, these are the types of phages that we could identify, and we've identified them by the bacteria that they can infect and kill. And we've been particularly interested in those that kill Bacteroides. That's our sort of favorite gut bacterium, if you like. And this bacteria has been associated, again, with health benefits. And we have seen that these Bacteroides can be depleted in ME patients and other diseases. So we're particularly interested in isolating viruses that can infect and kill this type of bacteria. And so this is one of the avenues of approach that we're taking. We're trying to isolate the virus. We can isolate them. We can sequence their genomes. We can put it in a database so they can be used to identify other viruses. This one in particular, this is what they look like, against Podoviridae. And this we know can kill certain species of Bacteroides. And so we're developing assays now to look at whether or not, if we put these viruses in with a normal healthy microbiome, does it kill the Bacteroides to give the dysbiosis that we see in ME patients. So this is just one virus. We're in the process of trying to isolate many more. And so this is all very nice. You know, we can clearly use genomics to identify viruses and bacteria. They can give us some indication of the function, but they can't really tell us for sure that this is what that bacterial virus does. So for that, we have to focus down here on what these bacteria and viruses produce or cause to be produced, the metabolites. And we've heard already about metabolomics. There's been some studies that have been done on metabolomics in less severely affected ME patients, the mild moderate there. And clearly, there's evidence from these patients there's altered energy and lipid metabolism. So that was our starting point, or Daniel's starting point, to look at the metabolome in severely affected ME patients, as I said. And also, none of these studies really included the same household healthy control. So again, it's an additional control that we have in our analysis. And I have to say the results have been somewhat disappointing in that we've not been able to replicate the results from the mild-moderate patients. So despite us being able to look at virtually all the same metabolites that others have done in less affected ME patients, we failed to detect any significant differences, altered lactate, short-chain fatty acids, etc., or amino acid catabolism. And then looking at the feces, the fecal microbiomes, this is more closely attuned to what your microbiota is producing. Again, there's nothing really that jumps out there as saying this is a signature of the fecal microbiota with severely affected ME patients. We did identify one difference here. It's a tentative identification. That's why it's in the parentheses. This is interesting because it's a bile acid, and bile acids are important in lipid metabolism. And some patients stood out from others in this group in that there were some differences associated with carnitine metabolism and the generation of TMAO, which has been linked to both health as well as disease. So nothing really jumped out from that. So in summary, our analysis, like those before, have clearly identified changes in gut bacterial populations in severely ME patients, but they may be distinct from the descriptions in less severely affected patients. The virome is different in ME patients. We don't know too much about that yet in terms of how it's different because of the issues with the being able to identify the viruses, but we're, so by isolating them, we're adding to the databases, so I think as time goes by, we'll identify more and more of these viruses. And our analysis of the metabolome shows that it doesn't sort of correspond to that described for mild moderate patients. So the statement at the bottom is something that's been mentioned by Oyston yesterday in the conference, and maybe he'll mention it again, that maybe what we're looking at is different diseases. Severely affected ME may be a different disease to that is present in mild or moderately affected patients. And so the biochemistry, physiology, and pathophysiology may be distinct. So we have to look at these patients as if they have maybe a different disease. That's the hypothesis. That's something we're hoping to be able to test more thoroughly as time goes by. So in the last part of my talk, the last few minutes, I just want to talk about some new initiatives. So this is what we're doing now and what we're hoping to do in the future. And there are three things I'd like to talk to you about. One is to do with GP training. Okay, I think we're all aware that GPs know very little or anything about ME. And there's a real need to try and educate them or re-educate them. I'll talk about a joint initiative that's part of the eMERGE structure that Rich has described, and then some clinical trials that we're getting involved in. So this is the GP scheme, and this was set up by one of the regional health authorities in Norfolk, the Norfolk and Waveney Clinical Commissioning Group. And with support from the Invest in Me charity and the Quadram, we've agreed to co-fund this. And this will allow a GP to spend at least 1 day a week at the Quadram getting involved in research for a year. And the aim is that this will improve the quality of care eventually for ME patients. They'll go back to the practice and serve as a champion of ME CFS research. And it will also keep GPs up to date, more up to date with actually the research that's going on in ME. So this position is currently being advertised. I've been told by the Norfolk and Waveney CCG there are people out there looking for this sort of opportunity. So I think this might be something that we can build on in the future and get more GPs aware in the UK at least, or maybe in Norfolk, about what ME is and how we can investigate it. The second is a joint PhD studentship that Invest in ME and the Swedish ME charity are with the University of Anglia co-funding. The PIs here, so Jonas Bergqvist, who's known to most of you, I'm sure, and Penny Powell from UEA. This will be advertised in the summer, and we're hoping to get a student, perhaps a Swedish student, that will register at UEA and spend time both in the Quadram and UEA, as well as Uppsala. So I think that's a very interesting development in terms of building cooperation across European countries for ME research. And then the last one is bacteria therapy. So based on the abnormal or dysbiotic gut microbiota, what can we do to try and restore a healthy microbiota in ME patients? And this can be looked at from the context of lawn care. Okay? So here we have a flourishing lawn. Here we have one that's infested with weeds or is depleted of nutrients. And the problem is that weeds can outgrow, and we need to kill off the weeds. So there are three approaches we can take. One is we can put down new seed, which is probiotics, living organisms. The other is we can put down turf food, or prebiotics, and that's the food that you eat to promote the growth of beneficial microbes. Or you can take the more drastic approach and replace the lawn. So probiotics, I think evidence to date suggests there's very little evidence that they're effective in ME. So we are looking at the more drastic approach, which is replace the lawn. And for the gut, this is microbiota replacement therapy, or FMT, fecal microbiota transplantation. Now, despite what you may have read recently, this is not something that's new. It's been around for a very long time. The Chinese have been using this for centuries, back in the 4th century. And in the 16th century, a Chinese physician developed something called yellow dragon soup, which was used to treat a whole array of not just GI symptoms, but other symptoms. Since then, we've moved on. So in the Second World War, German soldiers in East Africa were overwhelmed by dysentery, and somebody noticed that the local people would follow camels, collect their stool, and use that in various foods to consume that to protect them against dysentery. That led German scientists to develop this ingredient, Bacterial subtil, which can still be bought, and is essentially derived from camel's poo, or the microbes in the camel poo that protected these soldiers from dysentery. Since then, 1957, Stanley Fecal fed surgical patients with their own encapsulated feces in the ersatz trial. And then perhaps more prominently, in 1958, Ben Eisman used fecal enemas to cure 4 patients at near death from a form of colitis. All 4 patients were cured with this treatment. Since then, Thomas Borody in Australia has, since 1989, been using FMT to treat a whole array of disorders. And then in mice, we can show we can transfer obesity by just transferring microbes from a fat mouse to a thin mouse. And then really the thing that's made an impact in terms of clinical care is the work from Els van Nood in the Holland, where they used FMT to cure patients with Clostridium difficile infection. This is now part of the NHS regimen for treating C. difficile in the UK, and I think elsewhere as well. So in terms of the future, we can imagine various diseases which might be amenable to this sort of therapy, including ME. Thomas Borody has reported, way back in 2000, successful treatment of ME patients, 60 with mild to moderate, 52 of which had a complicating GI issue, like irritable bowel syndrome. His FMT was a mixture of 13 microbes, and it was administered via transcolonic infusion or a rectal infusion. 42 responded in terms of their fatigue, sleep deprivation, lethargy, and strikingly, 7 out of 12 were symptom-free 15 to 20 years at follow-up. And resolution of GI symptoms was seen in around 80 or 90% of the patients. So these are all individual case studies. What we really need to do in order to test the effectiveness and safety of FMT is a clinical trial. And so there are 2 trials that I'll talk about. One that's currently underway in Norway, Dr. Peter Holger Jonsson. This is an interventional clinical trial. It's randomized, and there's a placebo comparator as well. The FMT is delivered via an enema bag, and you can see from the table here the types of analysis that have been conducted. This involves Quadra. We are doing microbiome analysis. I think Maureen's group is looking at intestinal permeability, and the Umeå University in Sweden is also involved in some of this analysis. And the outcome measures are here. They are mainly subjective outcome measures based on patient questionnaires, improvement in quality of life, the fatigue severity scale, etc. And this is now underway, so it's currently up and running in North Norway. Close to home, we're proposing to undertake a clinical trial in Norwich called the Restore Me, and this is designed to determine the efficacy and safety of a fecal microbiome transplant in ME patients. We are looking to obtain evidence of patient acceptance, safety, and importantly, efficacy as well. And we are measuring using both subjective and objective measurements to assess the outcome. And we are also incorporating various research questions to look at how the FMT might actually be working or not working, as the case may be. So this is the study design. It's a single-site, randomized, double-blinded, placebo-controlled study. We're looking to recruit around 80 patients. This will be through the St. Helier chronic fatigue syndrome. Amalak Banzel is going to be in charge of that. We'll divide the group of patients up into 2. One will receive the treatment FMT, the other will be the placebo. And we're using the results from the comeback study, the Norwegian study, to improve the design as we get closer to starting our own. Our study is different from the Norwegian one in that the FMT will be delivered by a nasojejunal tube, not an enema. We have objective outcome measures, the most important of which are that we are measuring physical activity via various devices, and I've been talking to people at the conference here about what sort of devices these might be. There's also walking tests, and we're also very interested in cognitive function, so we're doing some very in-depth analysis of cognitive function. Gut, brain. Is there any improvement through the FMT in cognitive function in ME patients? And blood samples will be used to look at the battery of sort of immune parameters that have been described by others. So these are the primary endpoints as well as secondary endpoints. Is there an improvement in maximum daily activity using maybe the Vivaro or the Actigraph? watches, the 6-minute walking test, the DSQ-2 score, improvement in pre-intervention, so particularly those patients that present with GI symptoms. Then the secondary endpoints relate to change in cognitive function, etc. So in terms of where we are, so unfortunately for us, the UK Medicine and Healthcare Products Regulatory Agency, the MHRA, has said for any other application other than C. difficile infection, FMT is to be treated as a medicine. All medicinal products therefore need to be produced according to the principles of GMP, and they need a license to be able to do this. So we are having to equip a laboratory at the Quadra that meets these requirements. You can see where we are from the timeline here. We're about due for an MHR inspection, which if we pass, we then can use the room to prepare the FMT. We obtain the license, hopefully in advance of this. Then we can apply for the license and ethical approval to undertake the clinical trial. And again, this will require a considerable amount of time to be considered and refined by the regulatory bodies. Then we'd hope to recruit patients sometime for this time for a start date next year. So this will all cost a lot of money. Invest in ME have been very generous in providing us with pretty much 50% of the money that we need to undertake the trial. So we have a window of opportunity now to try and raise the additional funds. And for that, we need all your help and hopefully donations to help this trial come about. So my last slide really is to thank the people that have supported our research. Solve ME, CFS Initiative has provided support for Fiona's project, the Virome. Invest in ME Research has sponsored 3 PhD students, all with the aim of trying to develop the Quadra as a centre of excellence for ME. And last, but by no means least, are the patients and the carers that continue to provide us with support and samples for our research. Thank you very much.